Alright, here's chapter 4.3, and it's going to focus completely on the fungi. Here are 4 objectives that will be required for you to know for this section. Listing 2 detrimental and 2 beneficial activities of fungi from the viewpoint of the human. What are the 3 main forms of fungi? Where is chitin found? And then know the following terms: heterotrophic, saprobe, parasitic, hyphae, and mycelium. So there's quite a few different terms that you need to know. Alright, fungi. Alright, there's approximately 100,000 species of fungi, and sometimes people refer to that as fungi, but I like the fungi because they're kind of fun, as long as they're in the right place. So you have the macroscopic fungi, mushrooms, puffballs, gill fungi, those that you can see. Not necessarily see with the naked eye, because you can see mold and some yeast with the naked eye, but the macroscopic are going to be large entities. And mushrooms, there are some that are very good, some that are good to eat, some that are quite expensive because they are somewhat ornate or very rare. And then there's also different fungi, macroscopic fungi, that actually are quite toxic to humans. There are some that have psychedelic properties that you probably want to avoid. So if you are a person who likes to go hunt for their own fungi, you do need to be careful. And then there are the macroscopic fungi, the molds, the yeasts, and those are what we in microbiology really focus on, because those are the ones that are going to cause infection. Not that the macroscopic fungi can't cause disease, because they certainly can if consumed, but it is more of a toxicity issue versus an infection. Alright, there are 3 main forms of fungi, and that is going to be the unicellular form, there's going to be colonial form, and the complex or multicellular. So with the unicellular, you're looking really at the yeast form. The colonial, it's more of the mold. And then the complex or multicellular will be the mushrooms, the puffballs, the macroscopic. So those are going to be examples of the 3 types or the 3 forms of fungi that you can see. Here's a cross-section of fungal cell walls. So if you recall, bacteria in almost every case have a cell wall, whereas eukaryotic cells, some have cell walls, some don't. Plant have cell walls. Human cells do not have cell walls. But fungal cells that are within the realm of eukaryotic cells do have a cell wall. Alright, and here's the cell wall itself. Alright, and this is the interior of the cell as depicted with this arrow coming away from this cell here. You have the cell membrane, then there is this layer of chitin, there's glycoproteins, mixed glycan molecules that with the combination of these 3 contain or create the cell wall, and then the glycocalyx. And if you remember from the bacteria cells, this is kind of that slime layer, right? That sticky layer of carbohydrates and some proteins. And so this is why still some fungi have that kind of slippery, slimy feel to them. But the important thing to note is that fungal cell walls contain chitin. Right, so this is where you're going to find chitin in the cell wall of fungal cells. Alright, fungal cells have kind of 2 main forms, and one is going to be the yeast, and the yeast cells are round to oval shape, very similar to a coccus or a coccobacillus bacteria shape. They're going to replicate through asexual reproduction, and this process is budding. They're just simply going to create this small bud off the side of the parent cell, and it's going to break off. It's going to be much smaller than the parent cell, but eventually it will expand and grow to become almost identical to the parent cell. And then you have the hyphae cells, or the fungal cells that are hyphae. And these are going to be long, thread-like cells found in the bodies of filamentous fungi. So, versus yeast cells that are the little circular cells that you find in, skip that. The filamentous fungi is often what you see on bread, on fruit, and that fungus is generally furry-like, right? So that furry-like feature of fungus is because of the hyphae, those long thread-like cells. Now, an important word to remember is dimorphic. All right, so di is going to be two, morphic is going to be its shape. And so some fungal cells are considered dimorphic because they can take either form, the yeast or the hyphae form, depending on growth conditions. And this is typically going to focus on temperature and moisture. So if you have a cell in the environment, right, a fungus in the environment, it will often be the hyphae form because the environment is going to be somewhere around 25 degrees Celsius, 25 to 30 degrees Celsius, and it may or may not have a huge amount of moisture. If you think about an orange that's sitting on your kitchen counter, or a strawberry that's sitting in your refrigerator, right, that's not ideal living conditions in terms of pathogenicity for humans, but you can still have that fungus arise, and then you have to throw your fruit away. But if you take the fungus and put it into your body, right, now you have 37 degrees Celsius temperature, you have lots of moisture, you have lots of nutrition, and that hyphae fungus can then morph into the yeast form and then actually cause a very fulminant infection. All right, now, does that mean you're going to get sick if you eat a strawberry that has fungus growing or starting, and you eat one in the morning and you come back at night and the entire container is covered in fungus, the rest of the strawberries there? No, most likely not, because that isn't the type of fungus that's going to cause pathogenic or going to cause disease in humans. All right, only some cells can do this, some types of fungus. All right. Mycelium. All right, the different morphology of fungi. These words I don't think are on your list of words to memorize for the exam. Mycelium is. All right, so mycelium is the woven, intertwining mass of hyphae that makes up the body or the colony of the mold. So this is what you would see on a strawberry when you just look at the strawberry and you see the gray fur on it. Right? All of that is the mycelium. And you can look close at it and you can see the individual hair-like parts of it. That's the hyphae. All right, now a different way to classify fungi is whether or not they have septa or if they are non-septate. And so the septa are going to be cross walls found in many fungi, and this causes the hyphae to look like a long string of cells, and we'll see a picture of it on the next slide. But then there's non-septate hyphae that don't have that cross wall, and so then it looks like just one giant long cell. The vegetative hyphae are responsible for the visible mass of growth. So that's what you see on the surface of fruit, bread, your petri dish. And then there are the reproductive or fertile hyphae. And these are typically going to see at the end of the hyphae, or at the end of the filament. And those are going to produce spores, which will then allow for movement of the spores for reproduction. All right, so here is the hyphae structure. This is the mycelium. All right, so this is a large mass of individual hyphae. All right, so each string here is the hyphae. And then you have the septum, which you can see is that tiny line right there. But this is a cartoon diagram, and this is septate hyphae, where you have the cross wall. Right, cross wall here, here, here. And each section is going to have its own nucleus. The non-septate hyphae don't have the cross wall, and so it just looks like one continuous filament, and then you will randomly find nuclei throughout that. Right, it may not be, it doesn't look, it's kind of hard to explain. With the septate, you will generally always have one nucleus in each section. With the non-septate, you just won't have as many nuclei. You'll have one filament that you can look down and stain and see, 3 or 4 nuclei that help to maintain the function of that non-septate hyphae. All right, so here's a microscopic look at yeast. Right, the cell is very similar to what you saw at the very beginning of the chapter with the basic eukaryotic cell structure. This is the parent cell, and then this is a new cell forming through that budding process. And everything is smaller scale compared to the parent cell. But once this bud breaks off, it's going to be its own cell, and it will eventually expand to be the same size as the parent cell. And what's interesting is each time the parent cell gives off a new cell, it leaves a bud scar. And if you look at this on the microscope slide, you can actually see that. And this is really what it looks like. It's pretty cool. And it's actually pretty cool to see this process on the microscope slide. Now, with yeast, you can sometimes have pseudohyphae, and it's fake hyphae because you have this string of cells that are being produced, but this string of cells is being produced because the new buds simply are not breaking off, and so you have this fake chain of cells. They're normally not in chains. All right, just a couple more slides here. With fungus and human disease, you have a couple of different terms. We'll start at the bottom. Mycoses are going to be fungal infections. All right, just simply infections caused by fungal organisms. You can have a primary pathogen. That organism can sicken any healthy person. And then you have opportunistic pathogens that make individuals who are immune compromised or weakened in some way sick if they become exposed to that. Whereas the opportunistic pathogen, generally people who are healthy, will not be at risk for those diseases. All right, some words here that you do need to know. Heterotrophic, saprobic, parasitic. These are just the ways that fungal organisms can be classified according to how they get nutrition, and you can read through those. But it's interesting to note that fungal organisms can actually thrive in nutritionally poor or adverse environments. It's just interesting that the way that they intake their nutrients allows for this. And that is the end of section 4.3.